Hi, my name is Lance Capel. This is Chem 530, Concepts of Biochemistry. And this is my presentation on the overview of glycolysis and gluconeogenesis pathways. So let's dive into the details of glycolysis and gluconeogenesis, and we are going to discuss the phases of glycolysis. All right, so let's get into the central importance of glucose. Glucose is an excellent fuel. It yields a good amount of energy upon oxidation. 2840 kilojoules moles of glucose can be efficiently stored in polymeric form, and many organisms and tissues can meet their energy needs on glucose only. So also, glucose is a versatile biochemical precursor. Many organisms can use glucose to generate all the amino acids, membrane lipids, nucleotides in DNA and RNA, cofactors needed for the metabolism. So for the four major pathways of glucose utilization, number one is storage. It can be stored in the polymeric form, which is starch, glycogen, and used for later energy needs. Second is energy production. This generates energy via oxidation of glucose and short-term energy needs. Third is the production of NADPH and pentoses, and this generates NADPH for use in relieving oxidative stress and synthesizing fatty acids, and it generates pentose phosphate for use in DNA RNA biosynthesis. And then the fourth major pathway is structural carbohydrate production. This is used for generation of alternate carbohydrates used in cell walls of bacteria, fungi, and also plants. All right, so let's get into glycolysis. The definition of glycolysis is it converts glucose into pyruvate, producing ATP and NADH. The location of glycolysis is in the cytoplasm of the cell. A little bit more in-depth definition of glycolysis. It is a 10-step metabolic pathway by which one molecule of glucose is converted into two molecules of pyruvate, generating small amounts of energy in the form of ATP and NADH. So next we're going to talk about the importance of glycolysis. Number one, it's almost universal. It's almost a universal central pathway of glucose catabolism. It's a sequence of enzyme catalyzed reactions by which glucose is converted into pyruvate. And also pyruvate can be further aerobically oxidized. Pyruvate can be used as a precursor in biosynthesis as well. And then some of the oxidation-free energy is captured by the synthesis of ATP and NADH. So the research of glycolysis played a large role in the development of modern biochemistry. Understanding the role of coenzymes, the discovery of the pivotal role of ATP, the development of methods for the enzyme purification, the inspiration for the next generations of biochemists as well. And also glycolysis breaks down glucose for energy, while gluconeogenesis synthesizes glucose from non-carbohydrates, which is both crucial for maintaining energy balance. All right, so let's get into the phases of glycolysis. The first phase is the preparatory phase. also known as the investment phase. So step 1, glucose is phosphorylated to glucose 6-phosphate by the hexokinase. And now in this step consumes 1 ATP. Step 2, the G6P is isomerized to fructose 6-phosphate by phosphoglucose isomerase. Step 3, the F6P is phosphorylated to fructose 1,6-biphosphate by the phosphofructokinase. And this step consumes another ATP. Step 4, the F1,6BP is split into 2 3-carbon sugars, the glyceraldehyde 3-phosphate and the dihydroxyetone phosphate by aldolase. The DHP is converted to G3P by triose phosphate isomerase. Now 2 molecules of G3P proceed through the pathway. And I have here the preparatory phase pathway on this image here. And okay, so let's talk a little bit more about the metabolic pathways. The biochemical reaction in the living cell metabolism are organized into metabolic pathways. The pathways have dedicated purposes. 1 is the extraction of energy. 2 is the storage of fuels. The synthesis of important building blocks. The pathways can be represented as a map. It follows the fate of metabolites and building blocks. Identify enzymes that act on these metabolites. Identify points and agents of regulation. And then also identify sources of metabolic diseases. And I have here a map of the metabolic pathways. Okay, so let's get into the payoff phase, which is the energy generation phase. So step 6, the G3P is oxidized and phosphorylated to 1,3-biphosphoglycerate by glyceraldehyde 3-phosphate dehydrogenase. This step produces NADH. Step 7, this is where the 1,3, the BGP donates a phosphate to ADP, forming ATP and the 3-phosphoglycerate by the phosphoglycerate kinase. And then step 8 here, the 3-PG is converted to 2-phosphoglycerate by phosphoglycerate mutase. And step 9, the 2-PG is dehydrated to phosphoenolpyruvate by enolase. And then lastly is step 10, the PEP donates a phosphate to ADP, forming the ATP and pyruvate by pyruvate kinase. Next here I have the net yield of first ATP, 2 ATP molecules per glucose, 4 produced, 2 are consumed. NADH, 2 NADH molecules per glucose, and pyruvate, 2 pyruvate molecules per glucose. All right, so next we're going to talk about the principles of regulation. The flow of metabolites through the pathways is regulated to maintain homeostasis. Homeostasis occurs when concentrations of metabolites are kept at a steady state in the body. When perturbed, a new steady state is achieved. Sometimes the level of required metabolites must be altered very rapidly, which requires the need to increase the capacity of glycolysis during action, and also the need to reduce the capacity of glycolysis after the action, and then also the need to increase the capacity of gluconeogenesis after successful action. All right, so next we're going to talk about gluconeogenesis. The definition of gluconeogenesis is it is the metabolic pathway that generates glucose from non-carbohydrate substrates like lactate, glycerol, and gluconeogenic amino acids. The location of this is mainly in the liver and then to a lesser extent in the kidneys as well. Okay, so let's talk about the first pathway, which is pyruvate to phosphoenolopyruvate. Pyruvate is converted to oxaloacetate by pyruvate carboxylase in the mitochondria, and this step requires ATP, so it requires energy. The oxaloacetate is converted to PEP by phosphoenolopyruvate carboxygenase, and this step requires GTP. Second is the reverse of glycolysis steps. So PEP is converted to 2-phosphoglycerate by enolase. The 2-PG is converted to 3-phosphoglycerate by phosphoglycerate mutase. And then the 3-PG is converted to 1,3 by phosphoglycerate kinase. And this step also consumes ATP. Okay. Also, the 1,3-BPG is converted to glyceraldehyde 3-phosphate by glyceraldehyde 3-phosphate dehydrogenase, and this step requires NADH. The G3P and the dihydroxytone phosphate are converted to fructose 1,6-biphosphate by aldolase. Next is the pyruvate to phosphoenolopyruvate. So 3-fructose 1,6-biphosphate to fructose 6-phosphate. The F1,6BP is converted to F6P by fructose 1,6-biphosphatase. Next, the F6P to glucose 6-phosphate is isomerized to glucose 6-phosphate by phosphoglucose isomerase. And then the G6P to glucose. G6P is converted to glucose by glucose 6-phosphatase in the endoplasmic reticulum. A couple of key points here. The gluconeogenesis requires energy. So 4 ATP, 2 GTP, and 2 NADH per glucose molecule. It is essentially the reverse of glycolysis, but it bypasses the irreversible steps of glycolysis using different enzymes. Okay, so a couple of key points here. So with gluconeogenesis, this requires energy. So it requires 4 ATP, 2 GTP, and then also 2 NADH per glucose molecule. So it's essentially the reverse of glycolysis, but it bypasses the irreversible steps of glycolysis using different enzymes. All right, so next we're going to talk about regulation. So glycolysis is regulated by key enzymes like hexokinase, phosphofructokinase, and pyruvate kinase. The gluconeogenesis is regulated by enzymes like pyruvate carboxylase, the phosphoenolopyruvate carboxygenase, fructose 1,6-biphosphatase, and glucose 6-phosphatase. So essentially, both pathways are reciprocally regulated to ensure that one is active while the other is inhibited, preventing a futile cycle. So these pathways are crucial for maintaining glucose homeostasis in the body, particularly during fasting or intense exercise when the demand for glucose is high. Okay, so we're going to get into some discussion questions. First, I'd like to have the students in the class divide into small groups of 3 or 4 students. And then first, I want you to tackle the comparison of pathways. How do glycolysis and gluconeogenesis differ in terms of their purpose and regulation within the cell? And then also, can you identify the key enzymes that are unique to gluconeogenesis but not present in glycolysis? And then next would be energy considerations. Discuss the energy yield of glycolysis. How does it compare to the energy cost of gluconeogenesis? Why is it energetically unfavorable for both pathways to be active simultaneously in the same cell? Okay, so next, the next set of discussion questions I'd like the class to work on is the regulation mechanisms. What are the main regulatory points in glycolysis and gluconeogenesis? How do hormones such as insulin and glucagon influence these pathways? And then I want you to discuss the clinical relevance. What are the potential implications of dysregulation in glycolysis and gluconeogenesis in metabolic diseases such as diabetes? Okay. And next, I want you to discuss the substrate utilization. What substrates can be used for gluconeogenesis, and what are the sources in the body? Also, how does the Cori cycle integrate with glycolysis and gluconeogenesis? And then we're going to get into some group activities for the class. First, we're going to, as a group, work on pathway mapping. So the activity for this would be I'm going to provide the students with blank pathway maps, have them work in groups to fill in the steps of glycolysis and gluconeogenesis, including intermediates, enzymes, and key regulatory points. The objective of this group activity is to reinforce the step-by-step processes of both pathway and also to highlight the key differences and regulatory steps. The next group activity is role-playing enzymes. So for this, I'd like to assign each student or a small group of students to act as a specific enzyme in either glycolysis or gluconeogenesis. I'd have them explain their role, regulation, and the significance. The objective of this group activity is to deepen the understanding of individual enzymes, functions, and also how they fit into the overall pathways. And the next group activity is presenting groups with a case study. So these case studies involve metabolic disorders. I'd have the students analyze which part of the glycolysis or gluconeogenesis pathway which might be affected and also propose potential treatments or interventions. The objective of this case study is to connect biochemical pathways to real-world medical scenarios, and then also to encourage critical thinking about how pathway disruptions can affect health. Okay. And the next group activity is a regulation simulation. I thought this would be a pretty interesting group activity as well. I'd use a simulation game where students can adjust levels of substrates, products, and regulators like ATP, ADP, insulin, glucagon, to see how these changes might impact the flux through glycolysis and gluconeogenesis. And then I'd also have the students debate on metabolic pathways. And for this activity, they would debate the importance of glycolysis versus gluconeogenesis under different conditions. For the regulation simulation, the objective of this is to visualize the dynamic regulation of metabolic pathways and also to understand the effects of different regulatory molecules. And then for the debate on metabolic pathways, the objective would be to foster critical thinking and also a deeper understanding of when and also why each pathway is utilized. And these questions and activities are all designed to help students understand the complex processes of glycolysis and gluconeogenesis by engaging them in interactive and critical thinking exercises. So just a quick recap, the location of gluconeogenesis is predominantly occurs in the liver and to a lesser extent in the kidneys. And the purpose of this is the metabolic pathway synthesizes glucose from non-carbohydrate precursors such as lactate, amino acids, and glycerol. And this ensures glucose availability during fasting or low carbohydrate intake. And then the reverse of glycolysis, gluconeogenesis mirrors the steps of glycolysis, but it operates with different enzymes that bypass the irreversible reactions of glycolysis. And then the key steps include the conversion of pyruvate to phosphoenolopyruvate, which consumes ATP and then also GTP, and the conversion of fructose 1,6 biphosphate to fructose 6-phosphate. And then the energy cost. Gluconeogenesis is energetically expensive, requiring 6 ATP equivalents per glucose molecule synthesized, and this reflects its critical role in maintaining glucose homeostasis. And then also regulation and integration. Hormones such as glucagon and insulin, as well as the substrate availability and metabolic state, tightly regulate gluconeogenesis, and it integrates with other metabolic pathways like the citric acid cycle and fatty acid metabolism to maintain overall energy balance. So glycolysis breaks down glucose to produce ATP and pyruvate, while gluconeogenesis synthesizes glucose from non-carbohydrate sources to maintain blood glucose levels. And these pathways are tightly regulated and crucial for cellular energy metabolism and also metabolic homeostasis in the body. And this is my presentation. I hope you enjoyed it. Thank you. And here is a list of all my references for my presentation. Thank you.